Hello and welcome to BIS103 Lecture 7. In this part of our lecture, in this video, what I want to focus on is a different topic, is gluconeogenesis. So our pathways to generate glucose from other resources. So we are deviating a bit here from our previous discussions on glycolysis, the PDH reaction and the TCA cycle that we are using to break down glucose and other sugars for generating chemical energy. So now we want to turn it around. We want to make sugars. And I'm bringing it here into the lecture at this time because we're actually using many of the same reactions just in reverse. So it's timely to discuss it at this point while the memory of all these reactions is still fresh. So our learning goals for this lecture part are that we want to describe the reactions of gluconeogenesis, specifically in its relationship to the three irreversible reactions of the glycolytic pathway. And we also want to be able to understand the compartmentalization of these pathways, comparing the gluconeogenic and the glycolytic pathway. So the big question really is, can we go backwards in the glycolytic pathway to make glucose? And the short answer would be no, at least it's not easy, right? Because we have three physiologically irreversible steps that we need to address. Right? You might remember this is our glycolytic pathways all the way through the 10 steps going this way. And here boxed in red are three irreversible reactions. The conversion of glucose into glucose 6-phosphate, our first kinase reaction using ATP to phosphorylate glucose. Then here reaction 3 from fructose 6-phosphate, the same kind of kinase reaction to fructose 1,6-bisphosphate. And our third reaction here, same theme, right? Coming from PEP using our high energy compound and a kinase reaction, now generating ATP to our pyruvate here. So these are our three physiologically irreversible reactions that we need to find a way to circumvent them. Otherwise, we cannot go backwards in the glycolytic pathway. So this is where gluconeogenesis, or GNG, comes in. It's our pathway to biosynthesize glucose from non-carbohydrate substrates. And I sort of put myself out here, and I would argue that GNG is a universal pathway that exists in all organisms. It exists in animals, in us. We are using it. It's actually critically important because even if we don't get glucose through our diet, our body really relies on specifically glucose. Specifically, the brain requires glucose for many of its functions. But also microbes are using it, especially those that live on non-carbohydrate food resources. And plants are using GNG too, for example, for germination and photosynthesis. And so when we talk about non-carbohydrate substrates, a few can come to mind. And I listed some of those here. The first one being lactate, right? This was our product of lactate fermentation. So this is one that might actually be very relevant to us. We can also use most of our amino acids, and we'll see an example on how we can use, for example, alanine for this purpose. Glycerol, coming out of the breakdown of fats, can also go through gluconeogenesis to produce glucose. And then fatty acids, again, coming from the breakdown of fats, are a special case. They can be used, but only in certain organisms. Yeast and plants actually can do this. Animals cannot. We do not have the enzymatic capacity to bring about the net conversion of fatty acids into glucose through gluconeogenesis. But these other substrates here, we are capable of doing. This is what gluconeogenesis looks like. I know it looks very confusing, but be assured we'll walk through it together step by step using two examples. So first, just for your orientation, right, we have here highlighted in blue the glycolytic pathway steps that we already discussed in our first lectures, right, coming from glucose, phosphorylation steps, rearrangements through our high energy compounds to PEP and then to pyruvate. Okay. So this is what we want to focus on. What we want to do now is go backwards, back to glucose. So our first example, right, could be lactate. We just mentioned lactate can be one of the substrates. So we could start here. If we want to see on how we could go back, right, our entry point in reverse would be pyruvate. And so lactate to pyruvate, that seems possible. That's our lactate dehydrogenase, right, from our lactate fermentation. That's a reversible reaction. So we can do this. We can go from lactate back to pyruvate. That looks all fine. One thing that you have to also keep in mind here, in addition to looking at the irreversible reactions, is your cofactor balance. Right? And this is important here too. Right? In this reaction here, going this way, we are using our NAD cofactor. We are generating NADH. Right? And this is a cofactor we will actually need in our reaction here, right? Our oxidative phosphorylation step that originally came from G3P to 1,3-bis PGA, right? If we want to go back in this reaction, we need the reduced cofactor NADH. So keep in mind, you must maintain cofactor balance. In this case, this is a given because through the reverse reaction from lactate to pyruvate, we're actually generating the cofactor we need. So we're good here. Just keep in mind, whatever you do, make sure you have cofactor balance, all right? So that's cool. We have made pyruvate. So far, so good. But now we are stuck here, right? Our reaction PEP to pyruvate is irreversible, so we have to find another way to do this. So let's walk through these steps of what we are actually doing here to bypass this step. And again, keep in mind, this is a lactate dehydrogenase, the LDH, right? Keep in mind from the previous lectures on fermentation if you're a bit shaky on the mechanism of this enzyme. All right, so what we actually do is we take pyruvate, currently in the cytosol, and we actually bring it into the mitochondria. So we're actually bringing it into a different compartment now to convert it. The first step we do is actually we take pyruvate now in the mitochondria and we convert it to OAA, right? We're using ATP, we're using bicarbonate to do so. This should ring a bell. This is actually the pyruvate carboxylase that we just discussed in the other lecture part in our anaplerotic reactions to refill the TCA cycle. So this actually is an enzyme that is doing double duty. It functions in refilling the TCA cycle and it functions in gluconeogenesis. Now we have made OAA in this reaction. The next enzyme is the PEP-CK or the PEP carboxykinase. Here we're actually using GTP to bring about the decarboxylation of OAA to generate our high energy compound PEP. Okay. And PEP now, we actually have a shuttle. We can transfer this across the membrane of the mitochondria. We can bring it in here. We have bypassed our irreversible reaction from PEP to pyruvate. Now we can feasibly go up, reverse in our glycolytic pathway. Let's look at these two reactions just for a recap in a little bit more detail again, right? This reaction we already had seen in the TCA refill reactions, our pyruvate carboxylase taking pyruvate, we're bringing in bicarbonate, right? And we're using our biotin cofactor that now, through the use of the NHL of ATP hydrolysis, can bind our bicarbonate here, and then the cofactor carrying our carbon dioxide group can be transferred over to pyruvate. We are generating oxaloacetate OAA here. The second reaction, we're using GTP here. We don't actually need a cofactor for this. We can take OAA and we can use the energy from the hydrolysis of GTP to bring about the decarboxylation down here at OAA and regenerate our enolic phosphate here, PEP. All right, so we have achieved our first step, right? We bypassed our first irreversible reactions here. We can now move up in the glycolytic pathway in reverse. We can do this step here because we have generated our needed cofactor, so that's fine. But now we're stuck here, right? In the original priming phase of glycolysis, where we have these kinase reactions to phosphorylate our glucoses, we have to go back in reverse. And the way we are doing this is we are helping ourselves by using phosphatases. So again, just sort of zoomed into these reactions now. Up here you see left to right, is our glycolytic pathways, right? Starting with glucose, kinase reaction to phosphorylate at C6 to glucose 6-phosphate. This reaction is fine, it's a reversible isomerization, that's not a problem for us. And then we have reaction 3 here, again an irreversible reaction, phosphorylation of fructose 6-phosphate at C1 to generate fructose 1,6 bisphosphate. Now, if you want to go back, we're using our phosphatases. So the first one being the fructose bisphosphatase. What it's doing is it's using water to break the bond between the oxygen and the phosphate here. We're releasing the phosphoryl group, dephosphorylating the molecule from fructose 1,6 bisphosphate back to fructose 6-phosphate. So we have overcome this irreversible kinase reaction by using a different enzyme, the phosphatase. Again, this step is fine, it's reversible isomerization. And then the next time we're using the same strategy here, we're using the glucose 6-phosphatase to dephosphorylate glucose 6-phosphate to free glucose. Okay. Same principle, but with one major importance here. Oftentimes gluconeogenesis, especially for the purpose of using glucose for energy production in other tissues and cells, you would stop here and use glucose 6-phosphate. Because there's no meaning in necessarily, in the same cell at least, go further and have to rephosphorylate. What is specific here is that this phosphatase actually occurs only in the liver. Right? Remember, our liver was the organ that is responsible for distributing free glucose through the blood system. And so this phosphatase is specific to the liver, is only occurring there to catalyze the step from glucose 6-phosphate to free glucose. If you want to sort of remind yourself how this sort of fits in with our world map of BIS103 and the glycolytic steps, have a look at our video on the BIS103 map part 1, where I explain this more in the context. And so here shown again, right, we want you to remember that the liver is responsible for generating glucose, free glucose to the bloodstream, mostly breaking down glycogen, whereas the muscle does not do this, right? The muscle is using it directly down glucose 6-phosphate for the production of chemical energy. That's why we have this phosphatase only in the liver, because really it's the liver's job to generate the free glucose for its distribution. All right, so that's how we can do it with lactate. Now let's have a look at another possible substrate, in this case, alanine. So another option that we have is if we're running out of glucose and we're running out of sugars in general, we can use other resources, one of which can be proteins. Proteins are made of amino acids, so you can break down the proteins into amino acids, one of which would be alanine, and you can use these amino acids now to generate glucose. It works a little bit differently, but we're helping us with many of the same enzymes. So let's have a look. First thing you see is this one here, right? We can convert alanine to pyruvate. This is actually a reaction we haven't looked at yet. We'll see it when we talk about amino acid metabolism. It's an aminotransferase. You don't have to worry about it quite yet. It's a fully reversible reaction, so it's fine. But one thing that you should pay attention to here is if you look at these molecules, right? Alanine essentially is a replacement of the carbonyl group here with an ammonia group. But this will also mean we cannot go directly back. So we have to do this aminotransferase reaction here. Okay. The next step then, you could argue, right? Why don't I just now go between lactate and pyruvate again? Right? There's something missing here that we have to pay crucial attention to, and that's our cofactor. Compared to our reaction from lactate back to pyruvate, we are not in this aminotransferase reaction here generating our reduced NADH cofactor that we need in our reaction up here. Right? So while this is reversible, we would get stuck in this point here if we do not find a way to regenerate our cofactor. So we have to go via a different route. The first step, or the second step, I should say, downstream of pyruvate, is again our pyruvate carboxylase. exactly the same reaction as we have just seen for gluconeogenesis from lactate, converting pyruvate to OAA. That's fine, that works, but again, we are not generating a cofactor. So now we are helping us with another reaction here. We are using OAA and we're converting it into malate, right? We're reducing it and we're taking the electrons from NADH into NAD. They're now going into malate, right? And malate can be transferred across a membrane into the cytosol. Apologies for that. Now malate here is in the cytosol now. We can do the reverse reaction here back to OAA. Okay, now we are oxidizing malate and we're reducing NAD into NADH. And now we have our NADH, the reduced electron carrier, that can go to our reaction up here, and we have our cofactor balance. Right? It's a complicated system just to make sure we have cofactor balance here. We're achieving it by using two isozymes, so two enzymes that have the same function, but in this particular case, have different compartmentalization, one here in the mitochondria and one here in the cytosol, both catalyzing reversible reaction, allowing us now to generate our cofactor. Next step is, again, familiar to us. We have our PEP-CK, our PEP carboxykinase, that we just used in our GNG coming from lactate, but it was in the mitochondria at the time. This is another example of isozymes. Here we have an isozyme that catalyzes the same reaction, converting OAA into PEP. But this is the cytosolic variant of this enzyme. Similarly here, using GTP to bring about the decarboxylation, we have achieved PEP. So we used a few more enzymes to overcome this first irreversible reaction from PEP to pyruvate, mostly because, or exclusively because, we needed to take care of bringing about the cofactor balance of having the reduced NADH cofactor that we need up here in our conversion of 1,3-bis PGA to G3P. Now, same thing again, we have made PEP, we can move up all the way here, and we can move up all the way here using the exact same phosphatases as I had shown you for the example of lactate. So now that we have seen two of these examples, we need to ask us now, we know how it works, where and when it occurs, right? Where do we do this? We already had seen it sort of moves between the cytosol and the mitochondria within the cell. And talking about where it works and when it works, we really have to talk about the Curies. They were a married couple, Gertie and Ferdinand Curie, and they really, in the early 19th century, were working hard and have developed a lot of the knowledge today of the gluconeogenesis concept and how it works. Gertie Curie actually got the Nobel Prize for this work, and 4 of her students that came out of her also won Nobel Prizes. So she really was one of the major pioneers of biochemistry in the understanding of sugar metabolism. So what did they develop? They actually developed this concept of the Curie cycle, thinking about how glucose and gluconeogenesis and glycolysis can work in concert for the distribution of glucose and our ability to generate chemical energy, right? So what we want to look at here is our bloodstream and then basically a liver cell and a muscle cell, okay? And so we already had discussed, right, our glycolytic pathway coming from glucose. For example, in the muscle, we can do glycolysis, we can bring it to pyruvate, generating chemical energy. If you're running out of oxygen, for example, so under anaerobic condition, then you would use your LDH enzyme, you would generate lactate in the muscle. That lactate could go into your bloodstream. You could transport it through the bloodstream and bring it into a liver cell, okay? The same then could also be true. We could use alanine to do the same system. We're showing it here for lactate because that's produced in the muscle as your fermentation product. Then in the liver cell, right, we can bring in lactate, or we can use other resources such as alanine. We can do GNG to glucose 6-phosphate. we use our phosphatase that is unique to the liver now to generate free glucose. That glucose can leave the liver cell, go back into the bloodstream, and back to the muscle cell. Right? So this is a Curie cycle, which gives you an idea on how we can maintain the function of a muscle, for example, if we're running out of oxygen, and how we can reutilize lactate for the production of glucose. Okay. So lactate is not just a waste product, it actually can be reutilized for the production of glucose. So when does this occur, and how does this potentially work, right? So you could imagine now different scenarios, and there are different outcomes of this Curie cycle under these different scenarios. So one that I want to present here is, for example, the excess of amino acids. So for example, you just had a big T-bone steak, you ate a lot of protein, you're bringing in a lot of amino acids, your body wants to deal with it, right? So in this case, right, you have eaten all the protein is broken down into amino acids, just abbreviated as AA here. You can bring it into a liver cell. This can now be converted, and we'll see some of these reactions later, into, for example, OAA, 3-PGA, pyruvate, and alpha-KG, so all glycolytic or TCA cycle intermediates. Okay. Those now can be used for GNG, right? We can bring them into GNG, we can generate glucose 6-phosphate. Okay. But what do we do now under these circumstances where we're bringing in a lot of dietary resources, right? In this case, because we're bringing in so much energy, we don't actually have a shortage. It is unlikely that we would make a lot of free glucose, unless you would have a glucose shortage at the time. But it's a less likely pathway that we would actually use our phosphatase to generate free glucose. Instead, most likely under these circumstances, much of this glucose 6-phosphate would actually be brought into the production of glycogen and just stored away for later. Another scenario, now the opposite. Imagine, right, you've been running all day, you didn't have anything to eat, you're actually running out of sugars, you're going into carbohydrate starvation. Now, what is happening now? You actually could get amino acids, now not from your T-bone steak, but you're actually starting to break down your own muscles. And so you're bringing in the amino acids, the same happens, right? We bring it into these different precursors, we're doing gluconeogenesis to glucose 6-phosphate. But obviously now under starvation conditions, you would not bring it into glycogen for storage. You will really largely now generate free glucose. You activate your liver glucose 6-phosphatase to generate glucose and bring this back into the bloodstream to support other systems. So this Curie cycle can work in different ways depending on the conditions of the living system, the living organism. The Curie cycle is actually sort of the foundational hypothesis that we have here, but research has actually been ongoing, and there are some new insights that actually show that it might be somewhat more complicated than the Curies originally found out. And so there are some new knowledge and new hypotheses that have come out in the recent years, one of which actually from George Brooks's group at UC Berkeley. So if you're interested, you can read up on his work. And him and his colleagues, they have a new hypothesis over the last years for which there's a lot of knowledge now that it actually seems to be correct. And they call it a lactate shuttle hypothesis. So you won't find that in your textbook quite yet, but I think it's worthwhile discussing on these new types of hypotheses that add on to our knowledge of the Curie cycle. So how does this lactate shuttle hypothesis now work, right? Now we're sort of picturing and we're honing into basically on only muscle tissue. So we're looking at essentially two different muscle cells here. Much like we had seen the Curie cycle, right? From the bloodstream, you could bring in your glucose into the muscle cell. You could do glycolysis to pyruvate. You want to use it up. You keep using it, right? You're running really long and fast. Your muscle is running out of oxygen. The cell is deprived of oxygen. In this case, right, you would do lactate fermentation, producing lactate. Before I had showed you in the Curie cycle, right, this would go back into the bloodstream, would go up to a liver cell, would go through gluconeogenesis, diverted into glucose, and that glucose would go back to the muscle. The lactate shuttle hypothesis now argues that it could also be possible that the lactate is directly transferred from a muscle cell that is deprived of oxygen to one that has oxygen, right? Not all your muscle cells at the same time are deprived of oxygen. So it could simply be transferred across to another muscle cell and could here be used, just as we had discussed earlier, to use gluconeogenesis from lactate to pyruvate, and then not up gluconeogenesis further. So we're only using a part of gluconeogenesis here, and then use pyruvate as we had discussed in our chemical energy metabolism, break it down to acetyl-CoA using our PDH, and bring it into the TCA cycle. So essentially, this lactate shuttle hypothesis argues that there might be a shortcut to the Curie cycle that is using lactate directly and only part of the gluconeogenesis pathway to very efficiently move energy around between muscle cells. And so industry actually already has picked up on this concept, and you find out there are some products here, some of these sport drinks, and this is not an advertisement for this particular one, just to highlight to you how it could be used, where if you actually look at the ingredients here, you don't have a bunch of glucose in there, you actually have alpha-L-polylactate. So instead of sugars, they are putting in lactate into the sports drink, as arguing that because of this lactate shuttle hypothesis, that it would be bringing energy to your muscle much, much faster than sugar would be in this case, right? So it's an interesting industrial application of this sort of rapidly developing knowledge. So this concludes our gluconeogenesis. The next lecture up will be on actually how we can produce our poly and disaccharides. So watch out for this video.